Welcome to BioVivacious. BioVivacious, as you know, is a YouTube channel dedicated to clear fundamental concepts of biosciences. Now let us discuss about the quaternary structure of a protein. 2/3 of all the proteins in a mammalian system has quaternary structure. What does that mean? It means that 2/3 of all the proteins that we have are made up of more than one monomeric unit. If there are two such units, we call it a dimeric protein, or if there are three, trimeric, tetrameric, etc. So now you can, based on your knowledge that you have acquired, you will be able to now see that there are many proteins that you already know has got more than one monomeric unit, and they are all having a quaternary structure. Now this quaternary structure is bound together very specifically by non-covalent interactions. You will not find covalent bonds in the quaternary structure. They are present only in the tertiary structure. In the quaternary structure is based on non-covalent interactions. There is a reason why proteins are made up of oligomers or more than one unit. One of the reasons why it is made up of oligomers, the first reason is it gives, if you look at the active site of the enzyme, if the active site is normally, it is between the two domains. You have one domain, you have one monomeric unit, in the second monomeric unit, they will come together. If the active site is formed normally between these two units, it gives a greater advantage. The advantage is, number one, it will give a greater surface area for binding with the substrate molecules. You will see that this is very clearly applicable in the case of DNA binding enzymes. It gives a greater surface area. And the second reason is you can regulate the enzymes, regulate the activity of the enzyme either through phosphorylation, dephosphorylation. The structure can move. That is another advantage. And the next advantage is you look at cooperativity that is seen in hemoglobin. Binding of a molecule at one site can regulate the activity at another site. You will see that binding of oxygen to one of the proteins, it will alter the structure at another site. So therefore, these are some of the reasons by which the oligomeric proteins have an advantage. The oligomeric protein have an advantage. Conventional biochemistry always used Greek alphabets, the smaller Greek alphabets like alpha, beta, etc. in order to denote the monomeric units. And along with alpha, the Roman numerals like 1, as a subscript it was used. Alpha 1, beta 1, it means that there are two such subunits, one alpha unit, one beta subunit. If beta 2, one alpha subunit and two beta subunits. Now there are also conventions where if the capital letter of English is used. You can use H, L, etc. You will see in antibodies, heavy chain and the light chain. There is also another convention based on where it is present. Is it present in muscle? Is it present in liver? Is it present in heart? Therefore, the concept of isoenzyme comes. Isoenzymes are enzymes with many such monomeric units joined together, and it will carry out a very specific function. And the monomeric units can have different sequences of amino acids, so it can give different mobility, and that is the principle of separating them by using electrophoresis. Let us look at a classic isoenzyme, LDH. There are many forms available. You can have an H4, you can have an H3M, you can have an H2N2. So many different forms of LDH is available in the system, carrying out different functions. What is important for us to remember is how these many forms will come together in order to carry out a function, and they are all joined together through non-covalent interaction that gives the quaternary structure of a protein. You will also see now that how in the different hierarchy existing. You have a primary structure, you have a secondary structure, you have a tertiary structure when it is a single polypeptide chain, and you have a quaternary structure when there are multiple chains available for a protein. When we talk about the quaternary structure of proteins, we should also speak a few words about the motion a protein undergoes. Remember, a globular protein is not static. They are constantly responding to the environment in which they are present. So depending on the environment, and there will be different types of motion in a protein structure. NMR, X-ray crystallography, all these are used in order to study the motion that is present in a protein. So based on these studies, 3 classes of motions has been detected. The first type of motion is the vibrations and the oscillations of atoms and very specific groups that are present in a protein. So look at the amplitude of this. It's about 0.2 nanometer, and they are very fast. Look at the time that it takes, 10 raised to minus 15 to 10 raised to minus 12 seconds. So at that speed they are very quick, and this is the first class of motion which occurs in a globular protein. And this is studied based on X-ray crystallography, so therefore you can imagine that it is happening in the solid structure, in the crystalline structure. In the second class of motion is called the concerted motion. So in the concerted motion of certain groups, certain residues, that it has an amplitude of 0.2 to 1 nanometer, and it will take a time lapse of 10 raised to minus 12 to 10 raised to minus 8 seconds. That is the second class of motion. In the third class of motion is the whole domain moving. Okay, so it can be the whole domain moving, opening and closing of the cleft. So you can correlate with the enzyme activity of a protein, or opening and closing of a channel so that molecules can pass through. This is the third kind of motion which is seen in the oligomeric proteins. So the amplitude is about 1 to 10 nanometer, and the time it takes to carry out this is roughly about 10 raised to minus 8 seconds. These are the different classes of motion which is seen in a protein. So it gives you an idea about how protein, it folds for a particular function to be carried out, and which are the type of motion which takes place within the structure of a protein. I'm sure you have enjoyed going through these videos on understanding various nuances of protein structure. Keep subscribing and keep watching.